Welcome back for the first of several short videos on protein techniques. So in this first video, in actually this whole series of videos, we are going to talk about recognizing and understanding how chemical properties can be used to separate proteins. And we're going to look at protein sequencing techniques. In this particular video, we're going to talk about centrifugation and salting out. So, couple definitions. So, the proteome is the functional representation of the genome. So, it is the entire set of proteins expressed in the cell. So, the proteome is all of those proteins. Now, the genome is an unvarying characteristic of the cell, but the proteome will vary from cell to cell. So if we want to purify proteins, we want to study those proteins, first we've got to get them out of the cell. So we have to disrupt the cell, and essentially we can grind it, blend it. We somehow need to break up those membranes, okay, to let everything free and intermix. We get what we call the homogenate, with the mixture of all the components of the cells, but no intact cells. We've broken all of those apart. And then if we want to separate them, we're going to use a centrifuge, and we're going to start at low speed. So we know centrifuge, some things will be pulled out. They'll form a solid pellet. We're going to look at a picture in just a second. And that first part will include kind of the bigger structures that are left when we do this at low speed. So for example, any intact nuclei would be maybe in that first pellet. And then it's going to have the supernatant, the stuff that's still dissolved. We're going to take that supernatant, centrifuge it again, okay, with a higher force. And now we're going to get another solid, another pellet. And so if we take a look at what this might look like, we have our original time, and we have both our pellet and our supernatant. And that pellet might have, say, for example, a bunch of nuclei. But then we're going to take just the supernatant and centrifuge it again, now at a higher speed. And now we're going to get maybe some mitochondria, some pieces like that, smaller things. Centrifuge it again, and we're going to have these soluble proteins left here, and we would get some other small pieces in that pellet. So essentially, we can start this way, separating out bigger items from smaller items. Okay, and then we want to really purify those proteins, and they can be purified based on many of their chemical and physical properties. So their solubility, their size, their charge, their binding affinity. Okay, lots of options. The first thing we want to talk about is kind of one of the simplest, just the idea of salting out. So differential solubility. Some proteins are more or less soluble than others. Okay, that salting out will be dependent on the overall charge, how soluble they are, overall charge of the protein, ionic strength, polarity. And the salt that we often use is ammonium sulfate. And we'll do this because it has a high molarity at saturation, means a lot of salt in kind of a low volume, which is important because we don't want to over dilute our solution trying to add all this salt, because diluting the solution makes purification harder. All right, it's low density, so we can easily use it in the centrifuge. Okay, let's look at a picture. Here we go. So let's identify what we're looking at. We have this whole solution. It has some proteins shown with the dark black circles. Those proteins will have a variety of charges, some positive, some negative. We've got a bunch of water, and we've now added our ammonium sulfate, shown in these red circles. So normally when a protein is dissolved, it is hydrolyzed. It's got all these water molecules around it, okay, keeping it in solution. But when we start adding tons of ammonium sulfate, those salt ions attract that water away from the protein. Okay, they have a greater charge density, more charge per area, and the water molecules are really attracted. Those polar water molecules really attracted. So the water molecules start coming over here to the ammonium sulfate, which leaves those protein molecules to interact with each other and pull together. And so they salt out, they form this solid. Okay, when enough salt is added, protein precipitates. And a positive of using this technique is that it doesn't denature the protein. So remember we talked about that secondary, that tertiary structure. We're not disrupting those with salting out. As you would expect, if you think about it, proteins that are more hydrophobic have more hydrophobic amino acids would salt out or precipitate earlier than ones that are really hydrophilic. Ones that are really hydrophilic have lots of hydrophilic amino acids. Those water molecules are going to be pretty attracted. It's going to take a lot of ammonium sulfate to pull them away. But the hydrophobic ones will salt out first. So think about what that means. That means that we could do this in multiple fractions, okay, fractionations. We could add some ammonium sulfate, see what precipitates, pull that out. Now we've got one sample of our protein, and now we can add more of the salt. Some more stuff will salt out. We'll get a new solid, and that was probably things that were a little more hydrophilic than the first set. Pull it out, add more salt, see what proteins. And so we can start separating proteins by pulling these out individually as we add more and more and more salt. If we look at an example purification table, so starting with the crude extract, so having just homogenized all those cells, and then doing the salt fractionation, we could measure the total protein. It's going down. We're always going to lose some with every single step, so that total protein is going to decrease. But if we have a specific protein we're looking for, we can measure for that, and here we go. We can see it's also decreasing. But the ratio, the specific activity, which is essentially the purity, is the stuff we're looking for, the protein we want to isolate, over the total protein that's there. That gives us the specific activity. And we see that it is increasing. So even though we're losing a lot of the protein in our sample, our specific activity is increasing. It's getting more and more and more pure. All right. Next up, we will talk about column chromatography.